Hi everyone, and welcome to our module on the HMP Shunt. The HMP Shunt is a series of reactions that is an offshoot of glycolysis, and it goes by several different names. I was taught to call it the hexose monophosphate shunt or the HMP Shunt. Some books will call it the pentose phosphate pathway, and it is also sometimes called the 6-phosphogluconate pathway. And the reason it's called a shunt is because glucose 6-phosphate is shunted away from glycolysis for this series of reactions. These are the molecules formed as glucose is converted to pyruvate for glycolysis, and the second intermediate is glucose 6-phosphate, and that is where molecules are shunted away for the HMP shunt. I've summarized on this slide the four most important things for you to know about the HMP shunt. First of all, you need to be aware that this series of reactions synthesizes NADPH, which has many uses inside of cells that we'll talk about in this module. This pathway also synthesizes ribose 5-phosphate molecules, and these molecules are very important for synthesis of nucleotides. There are two key clinical correlations of the HMP shunt that you should know. The most important and the most high yield is a deficiency of the enzyme glucose 6-phosphate dehydrogenase. We'll talk about this in detail. And then there's also a clinical correlation of thymine deficiency that relates to one of the enzymes in this pathway called transketolase, which we'll talk about in a minute. All of the reactions of the HMP shunt occur in the cytosol. The mitochondria has no role in the HMP shunt. And the reactions are traditionally divided up into two phases. The first series of reactions are called the oxidative reactions. These are irreversible and rate limiting, and these reactions convert molecules of glucose 6-phosphate into ribulose 5-phosphate, and in this process they generate NADPH. The second set of reactions in the HMP shunt are called the reductive reactions. These are all reversible, and these convert molecules of ribulose 5-phosphate back into fructose 6-phosphate, which can re-enter the glycolysis pathway. In doing so, they generate ribose 5-phosphate molecules, which can be used for nucleotide synthesis. So let's talk about the first half of the shunt first. These are the oxidative reactions. In this portion of the pathway, molecules of glucose 6-phosphate are converted into molecules of 6-phosphogluconolactone. And the enzyme that catalyzes this step is glucose 6-phosphate dehydrogenase. This is a very important enzyme because, as we will see, there is a characteristic clinical deficiency of this enzyme. As part of this first step, one molecule of NADPH is created. In the second step, a molecule of ribulose 5-phosphate is synthesized. This generates another molecule of NADPH, and importantly, a molecule of CO2 is removed, and that's because we are taking a 6-carbon structure of glucose and creating a 5-carbon structure of ribulose. The second half of the shunt involves the reductive reactions. These are all reversible reactions. It's very low yield to know the names of all the intermediates and all the enzymes. The important features of this portion of the HMP shunt are that molecules of ribose 5-phosphate are generated for nucleotide synthesis, and then one of the enzymes involved is called transketolase. This enzyme can lead to the formation of fructose 6-phosphate, which can re-enter the glycolysis pathway. There are a couple of things about the enzyme transketolase that you should be aware of, especially for step one of your boards. This enzyme transfers a carbon unit to create a molecule of fructose 6-phosphate. It requires thiamine, vitamin B1, as a cofactor, so patients who are thiamine deficient will have poor activity of this enzyme. In addition, this enzyme may be related to the Wernicke-Korsakoff syndrome. This is a cognitive syndrome that alcoholics develop when they are chronically deprived of the vitamin thiamine. Now, not all alcoholics who are deprived of thiamine develop the Wernicke-Korsakoff syndrome, and there are data suggesting abnormal transketolase enzymes may predispose alcoholics to develop this condition. The affected individuals may have abnormal binding of thiamine, and you should be aware of that for your board exams. Let's discuss the important products of this pathway for cells. The first one is the molecule ribose 5-phosphate. This is a picture of ribose 5-phosphate on the screen, and you can see that it almost looks like a nucleotide. We have a sugar and we have a phosphate group. All we're missing is a base right here. So if we add a base to this molecule, we would have either a purine like adenosine or guanosine, or we could synthesize a pyrimidine like cytosine, uridine, and thymidine. And in the modules on nucleotide synthesis, I talk about how these molecules from the HMP shunt are used to synthesize these nucleotides. NADPH stands for nicotinamide adenine dinucleotide phosphate, and it has a very similar structure to NADH, but don't confuse it with that molecule because it has an entirely different function. This is the structure of NADH on the left side of the screen here. On the right side of the screen is NADPH, and you can see that the only difference is the addition of a phosphate group to one of the ribose sugars. Unlike NADH, NADPH is not used for oxidative phosphorylation, and it is not used for the generation of ATP. NADPH is used in so-called reductive reactions. It will release its hydrogen to form NAD+, and in doing so, it will donate an electron to other molecules for chemical reactions. The first use it has is as a cofactor in fatty acid and steroid synthesis. So tissues like the liver and the mammary glands and the testes and the adrenal cortex, they all use a lot of NADPH in order to synthesize fatty acids and steroids. The second use is for phagocytosis, which we'll talk about in a minute. And then finally, it is very important for protecting cells, especially red cells, from oxidative damage. So let's talk about the utility of NADPH for killing of bacteria by phagocytes. This involves a process called the respiratory burst. Phagocytes generate hydrogen peroxide in order to kill bacteria. This is called oxygen-dependent killing. There is another process phagocytes use called oxygen-independent killing, and this involves using things like low pH and enzymes to destroy bacteria, but it does not involve oxygen. So for the oxygen-dependent killing, phagocytes need to generate hydrogen peroxide. In order to do that, they need three important enzymes: NADPH oxidase, superoxide dismutase, and myeloperoxidase. So this is the process of the respiratory burst. So you can imagine that a macrophage has engulfed a bacteria, and now it will use this series of reactions in order to kill the bacteria. The process begins with oxygen. That's why this is called oxygen-dependent killing. And the first step involves conversion of oxygen to an oxygen radical. This is done by the enzyme NADPH oxidase, and it uses a molecule of NADPH that comes from the HMP Shunt. The second step involves generation of hydrogen peroxide. This is done by an enzyme called superoxide dismutase. And then finally, the enzyme myeloperoxidase can use a chloride ion and create a molecule of hypochlorous acid. And you may know that hypochlorous acid is what you find in common household bleach. All three of these intermediates, the oxygen radicals, the hydrogen peroxide, and the hypochlorous acid, can all lead to bacterial cell death. They can kill the bacteria inside of the phagocyte. There is an important clinical correlation of the respiratory burst, and that is the condition chronic granulomatous disease. This occurs in people who have lost function of the enzyme NADPH oxidase, and therefore their phagocytes cannot generate hydrogen peroxide. If we go back a slide, you can see that NADPH oxidase is the enzyme that starts this whole process going. So people who are deficient in this enzyme cannot generate any of these molecules that lead to oxygen-dependent bacterial death. Interestingly, there are bacteria that lack an enzyme called catalase. These are called catalase-negative bacteria, and they generate their own hydrogen peroxide, and the phagocytes can steal or hijack the bacterial hydrogen peroxide and use it despite their enzyme deficiency. So if we go back a slide, in patients who lack NADPH oxidase with catalase negative bacteria, they can steal hydrogen peroxide from the bacteria, and then they can use that to kill the bacteria. In addition, they can convert it to hypochlorous acid and also use that to kill the bacteria. Catalase positive bacteria, however, break down hydrogen peroxide and do not generate any for the phagocyte to steal. Thus, the host cells have no hydrogen peroxide, and this leads to recurrent infections. There are 5 organisms that cause almost all of the chronic granulomatous disease infections. These are Staph aureus, Pseudomonas, Serratia, Nocardia, and Aspergillus. And all of these are catalase bacteria organisms, and therefore they don't generate any hydrogen peroxide for the cell to steal. Back to our slide of the utility of NADPH. The third use of NADPH is to protect cells from oxidative damage. And the easiest way to understand that is to talk about G6PD deficiency. If we go back to our overview of the oxidative reactions of the HMP shunt, the first step of this pathway is catalyzed by the enzyme glucose 6-phosphate dehydrogenase. So people who are deficient in this enzyme will have a reduced ability to generate NADPH by these two reactions. So the syndrome of a G6PD deficiency occurs because cells, especially red cells, lack NADPH. And NADPH is very important, particularly for red blood cells, to maintain normal function of the cell. It turns out that there are a number of environmental triggers which can generate hydrogen peroxide inside of red cells. These include things like infections, certain drugs, and fava beans. We'll talk about all of these in a minute. But the point here is that they lead to hydrogen peroxide formation inside of red cells. If the red cell has NADPH, then it can use that to degrade the hydrogen peroxide. We'll talk about exactly how in a minute. But in people who have a deficiency of the enzyme G6PD, they cannot generate enough NADPH, and this leads to hemolysis. And that is the hallmark of G6PD deficiency, is hemolysis in response to certain classic environmental triggers. So in order to understand how NADPH protects red blood cells from oxidative damage and hydrogen peroxide, we need to understand a molecule called glutathione. This is the structure of glutathione on the screen here, and it is very important for protecting red cells from oxidative damage. In order to understand this, I want you to imagine that we have a healthy red blood cell, but it is deficient in the enzyme G6PD. Now imagine that one of those triggers comes along and generates some hydrogen peroxide in the red cell. If glutathione is present, glutathione can convert the hydrogen peroxide into water, which is harmless. It does this through the enzyme glutathione peroxidase, and in the process, it generates a molecule of glutathione disulfide, which is basically two glutathione molecules linked by a sulfur bond. That's why it's called disulfide. The glutathione disulfide can then be regenerated back into glutathione by an enzyme called glutathione reductase, but this enzyme requires NADPH. It converts NADPH into NAD+. If this process occurs as it should, then glutathione is constantly regenerated and is available to defend the red cell against damage from hydrogen peroxide. However, for this process to work, you have to have a functioning HMP shunt, and for a functioning HMP shunt, you have to have G6PD. So if G6PD is deficient, then there will not be NADPH present, and glutathione cannot be regenerated, and thus the red cell will be vulnerable to damage from hydrogen peroxide. G6PD deficiency is an X-linked disorder, so in a board question, it's always going to be in a male. It's the most common human enzyme disorder, and this makes this a very high-yield subject for step 1 of your boards, also all your steps and the internal medicine boards as well. There's a high prevalence of the G6PD deficiency in Africa and also in Asia and the Mediterranean. There are data suggesting that deficiency of this enzyme may be protective against malaria, and that may be why it's common in places where malaria is also common. The classic presentation is recurrent hemolysis after exposure to a trigger, and the hemolysis may present as dark urine. So a male patient who presents with dark urine after eating fava beans is a classic way for this disorder to present. Interestingly, the other functions of the HMP shunt are usually okay. So as I told you before, the HMP shunt, in addition to generating NADPH, is also used for nucleic acids and fatty acid synthesis. Usually people with this deficiency do not have problems in these areas. The problem is solely with the red cells and protection from oxidative damage. There are two classic findings of a peripheral blood smear for a G6PD deficiency. The first one is called Heinz bodies, and the second one is called Bite cells. Heinz bodies are oxidized globin chains that have precipitated inside of the red cells. This is a drawing of what they might look like here. You can find actual smears online of what they look like. Bite cells are phagocytic removal by splenic macrophages of the Heinz bodies. So when these type of cells enter the spleen, the macrophages try to eat the Heinz bodies, and as a result, they generate red blood cells that have a bite taken out of them, and those are called bite cells. There are three classic triggers for hemolysis in G6PD deficient patients. The first classic trigger is an infection. Macrophages, as part of the inflammatory response, will generate free radicals, and these can be toxic to the red cells. Fava beans are also a well-described trigger. Fava beans contain oxidants, and if patients with G6PD consume these, they will develop hemolysis. And then the final important trigger are certain drugs. There are a number of classes of drugs which can cause hemolysis in these patients. The first group include antibiotics. Sulfa drugs are well-described triggers of hemolysis, and that's why I've bolded them. So is the rarely used antibiotic dapsone and another rarely used antibiotic called nitrofurantoin. And then finally, INH, which is isoniazid, is a tuberculosis drug, and it can trigger hemolysis. Several antimalarials can cause G6PD deficiency, especially primaquine and quinidine. And I told you that G6PD deficiency is common in places where malaria is common. So sometimes patients will be screened for a G6PD deficiency prior to starting therapy with one of these drugs. And then finally, aspirin and acetaminophen have been described to rarely cause hemolysis in patients who are G6PD deficient. You can diagnose a G6PD deficiency by doing something called a fluorescent spot test. This test detects the generation of NADPH from NADP molecules. It involves taking a spot of blood and putting it under UV light, and a positive test is when the blood fails to fluoresce under the UV light. And there really is no specific treatment for this disorder. Patients just need to be advised to avoid triggers, and they need to be aware that if they develop an infection, hemolysis may occur. And that concludes our module on the HMP shunt.